Welcome to the Great British Bioturbationathon. I'm here on the west coast of Scotland on a small island called Cumbrae. Just behind me in the Field Studies Centre, the Convet Seascape Survey team are busy preparing for 2 weeks of scientific research. They have to find and collect small little animals that live in and on the seafloor. These are called benthic invertebrates. So right now, I'm going to go and meet the team and head out to sea. The team spent their first day setting up an entire lab from scratch. In just 1 day's time, they hope to fill this with the animals they find from the seafloor. Local guide Duncan has worked in the Firth of Clyde for many years and has a good idea of where they might find these benthic invertebrates. Collecting samples from the seafloor is no easy task, and this project requires a combination of local knowledge, innovation, and pure grit to get the job done. Hi Duncan.
Hi.
I was just wondering, how are you finding these areas to sample?
Well, if you look at the charts, you can see on the charts there's various marks on it. This one we're heading to, it says SOM, which is short for soft mud, hopefully full of animals.
So we're out here on the boat on the first day of the Great British Bioturbationathon. This is the grab sample, and it's just gone down to grab a big clumpy bit of the seafloor. Here they're flushing it with seawater to loosen up all the particles. Now pouring it over a sieve to filter out the big and the small particles. Pushing in all that globulous mud. All the mud is falling to the bucket, and then species are being revealed one at a time.
Female?
It's a sea mouse. Wow. Wait, so is this a worm?
It is.
Oh wow.
So now you can see all the segments that shows you as a polychaete and its different bristles.
Look at the colours, it's like glowing. That is actually so cool. So while all this chaos is going on, Naomi has been beautifully sat here doing some incredible drawings. Naomi, tell us a little bit what you're doing today.
So at the moment I'm just sketching what's going on, so it's all quite quick fire. So they're really rapid sketches of just the people and some of the activities, and trying to get a little bit of a sense of how the work's carried out, how the research happens.
So how do you think your artwork can add an extra element of communicating this science?
An artist maybe has a slightly different perspective on things, and maybe it communicates it to a different audience. So I'm painting with seawater here as well, so using the mud, and hopefully I'm going to be using the colours from the mud to make some more work later on.
The team needs 5 individuals of a single species if they want to include that species in the experiment. The more grabs the better, so it's all hands on deck to get the job done.
Well, this is an absolutely fantastic spot for us to come for the questions we're interested in, but most importantly, we can find a big range of benthic species. We're getting there, and certainly we're getting some very interesting species, and that's the most important thing.
Yeah, I mean, it's always chaotic working on a boat. You know, the winch semi broke earlier. Pulling mud out of the deep is always difficult, but it's good fun and we're all having a good time. So
overall, the day was very productive. We managed to do a lot of grabs, about 40 to 50 grabs in total over the course of 5 to 6 hours. So now we're going to take the samples that we have, take them up to the lab, put them in tanks, and get ready for experiments in the upcoming weeks.
The team will spend the evening setting up their lab to make the environment suitable for the species they'd caught. By the morning, it was barely recognisable.
When we include the topographical variation in the sea floor, actually the sea floor covers about 97% of the Earth's surface. And within that, 27 of the 28 major phyla, or groups of organisms, are included, many of which are unique to that habitat. And actually we know very little about what each of those are doing individually.
Well, what we need is 5 individuals of every species that we have. We need our experiment to be replicated. So we did quite a lot of identifying the species to make sure we had 5 of the same. And then basically we were allocating them into our experimental tanks.
So what we're doing is we're collecting those animals when we're measuring their ability to mix mud. And we're hoping then to relate that to how they move nutrients up and down and carbon up and down within that top 10 to 15 centimetres of mud. Because what we want to understand is how that relates to long-term burial of carbon, what we call sequestration, where the carbon is locked away for the long term.
Hello.
Hi.
Martin, what are you up to?
I'm just looking at one of the species we collected yesterday. If you want to come in and I can show you around.
Wow, there's a lot of tanks here. How many different species did you find?
So yesterday we were at about 20 species at the moment, and we've collected all those and put them into the tanks behind me. These are the individual species, so we've got different sized aquaria depending on the size of the organism. We keep them here in this water bath, and the water bath's chilled to kind of the temperatures that are out at sea. Because these species are from depth, we also need to keep them under blue light.
So you've got a muddy layer you've put in,
Yeah.
and then what is this glowing yellow sand on the top?
So these are the way we can visualize what these animals do. The primary mechanisms in terms of their behavior is to mix particles. Scientifically we call this a luminophore, but it's really just dyed sediment particles, and they're dyed with a dye that fluoresces in ultraviolet light. So what we can do is we can add this, but as the animals work these in through the sediment, they become more dispersed, and we can use photography to look at how those particles are dispersed and characterize what those individual species do. Those activities influence, nutrient cycles, the carbon cycle, and also the structure of the sediment.
What we want to do is to find some of those really good bioturbators, but also some of the not so good bioturbators, and then take them back to our research aquarium at Exeter University, where we can label the carbon and actually then follow the carbons. We can't do that with a huge number of species. So by picking our champions, we'll be doing that work on the right species.
Some species are large that have a very low effect, but some species are very small but can have quite a large effect. And that's what's exciting about this program, is that we're kind of breaking expectations on what species do. So most of the biodiversity in the oceans is actually in the sediments. So these are kind of levels that rival the rainforests and the coral reefs. And the seafloor environment is arguably the largest habitat on Earth, covering about 97% of the planet when you include the topographical variation. But clearly changes at that kind of level are going to have a huge effect on major biogeochemical cycles and the carbon cycle.
What's going on behind me will continue for another week whilst the scientists select the key bioturbation champions and then take them down to the University of Exeter for further analysis. Watch our next video to see which these species are, and stay tuned on our social media channels.